Hello, I'm Kenny. I'm a clinical scientist and I work here at the Southwest Genomics Laboratory Hub. It's here that we provide genetic testing for patients with rare disease and cancer in the southwest of England. Genetic testing can be undertaken for a range of different reasons. It could be to confirm a diagnosis. It could be in a predictive setting where an individual is at risk of a hereditary condition. It could be to monitor disease. It could be to identify individuals who respond to particular types of therapy, or it could be a pharmacogenomics test. Pharmacogenomics is the study of the relationship between genes and genetic variants and how they influence someone's ability to metabolize a drug. DPYD gene analysis is an example of a pharmacogenomics test, and the Southwest GLH has recently launched a rapid service for testing this gene. Today, we're going to take you for a look inside the laboratory and show you what's involved in the testing procedure. But before we do that, one of our local consultant oncologists is going to explain why the test is important and why the results are required so quickly.
So hello, my name's Steve Falk. I'm one of the oncologists in Bristol, and we're going to talk today about DPD testing and why we do it and how it's done. And why does it matter? Because we use very commonly in chemotherapy a drug called 5-fluorouracil and its tablet version called capecitabine. We know that there's a small proportion of the population that do not metabolize these drugs correctly, and that's due to inherited changes, which normally doesn't make any difference to you as an individual. But when you get these chemotherapy drugs, can make a very big difference and result in life-threatening side effects. So what we can now do is test for this deficiency and therefore change the doses of the drugs and improve the safety for patients. And what's that meant in practice is that actually far fewer patients are getting really sick with the chemotherapy drugs that we have, and far fewer are coming into the ward. So this DPD testing is now a mandatory part of testing patients before they start chemotherapy with these drugs.
Samples from across the southwest of England arrive at the Severn Pathology building throughout the day. Samples are received at the main specimen reception, where they are unpacked and sorted based on which department undertakes the specified test. About 100 samples are expected every week for DPYD gene analysis. The laboratory has a telelift system to move samples between the main specimen reception and the respective departments within pathology. Let's follow a DPYD sample up to the genetics department. A technician receives test referrals by booking them into the laboratory database and then logs the required test and appropriate sample preparation procedures. The Genomics Laboratory Hub and the wider Genomics Medicine Service provide hundreds of different genetic tests from the National Genomics Test Directory. Once the sample is booked in, labels are printed with the patient's name and sample number, and the blood sample is then moved to the extraction laboratory where it is prepared for testing. Most molecular genetic tests are undertaken on DNA, which is extracted from a blood sample. There are a range of different methods of DNA extraction available, some of which can take several hours to complete, and the most appropriate method to use depends on several factors, including the technology which is used for the test. The technology we use for the rapid DPYD service uses loop-mediated isothermal amplification, or LAMP for short, with melt curve analysis, and requires only a very small amount of DNA. Sample preparation is very simple, requiring only a single drop of blood to be added to a lysis buffer. This bursts the white blood cells, releasing their DNA into solution. Once this is completed, the technician takes the sample round to the pre-amplification laboratory, where another technician sets up the LAMP reaction. The technical work for the LAMP reaction is very simple, requiring only 2 liquid handling steps per well. The technician dispenses a specific reagent into each well of the reaction plate to test for one of the 4 DPYD variants. They then add the patient sample to each of the wells and mix the 2 solutions together. The reaction plate is sealed and then taken to the amplification laboratory, where they load it onto a real-time analyzer. Variations of polymerase chain reaction, also known as PCR, underpin most of the work that's undertaken in a genetics laboratory. Unlike PCR, which requires thermocycling to amplify DNA and can take several hours to complete, lamp-based amplification is carried out at a single temperature and together with melt curve analysis is completed in less than 1 hour. As the melting temperature is gradually increased, the reporter molecule is denatured from the template strand and a fluorescent signal is released. The specific temperature at which this occurs identifies the nucleotide at that position. So the shape and position of the melt curve tells us the genotype at that position. In the first 3 wells, the melt curve has a single peak indicating the patient is homozygous for the normal variant at the respective position in the gene. In the 4th well, the melt curve has 2 peaks, indicating the patient has one DPYD gene with the normal variant and the other DPYD gene has the mutant variant, i.e. they are heterozygous for the variant C.1236G to A. The presence of this variant indicates that this patient is at increased risk of fluoropyrimidine toxicity and the dose should be reduced accordingly. A report is written by the scientist and emailed to the referring clinician.
So each day the results come back and they're sent through NHSnet by email, and we have a generic inbox within the trust where all the results come so that they can easily be accessed by our members of staff, because all our chemotherapy prescriptions are double checked by pharmacy before they're dispensed. And as we can see for this patient, they've got a DYPD variant and a gene abnormality that means that actually they're at significantly greater risk of side effects from our chemotherapy drugs. And the result we get back tells us, one, in nice big letters, there's an increased risk of toxicity, and it helpfully tells us what the variant is, but also importantly what we need to do about it. So all members of staff can easily see that, access it online with our trust systems, know that we need to reduce the dose and get that all done and sorted before the patient starts treatment. And indeed, in our chemotherapy prescribing system, we have a DPD line that has to be completed before the prescription can be given. And this is all about making sure that we keep our patients safe.
I hope this film has proved a useful insight into the DPYD service here at the Southwest Genomics Laboratory Hub. If you'd like any further information, it's available on our website.